Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Next Generation Platforms for Strain Optimization, Reducing the Cost and Time for Commercializing Novel Biological Molecules, and our sponsor is Twist Bioscience. Our panelists today are Dr. Sunil Chandran, Vice President of Research and Development at Amaris, and Jason Downing, Director of Product Management at Twist Bioscience. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of the screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you don't see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentations have concluded. Dr. Chandran, please go ahead.
Hello, everyone. Let me just start by sharing my screen here. Great. Okay, so hello, everyone. First of all, thank you, Christy, for the introduction. I would also like to thank the Twist team for this opportunity to talk about the Amaris technology platform. Today, I'll be presenting the journey that Amaris has taken from a company that started with the goal of producing a reliable supply of a key antimalarial drug to a company that currently has its products in more than 2,000 brands across the world. So this first slide encapsulates what Amaris does at a high level. We engineer microbes that consume sugar and convert it to pure ingredients. Our goal is to make clean manufacturing powered by biology the industry standard for how all product ingredients are manufactured in the future. Amaris was founded based on a project that started at UC Berkeley in Jay Kiesling's lab, and it was a project to develop a fermentation-derived source of the antimalarial drug artemisinin. Until Amaris came along, the only source of this drug was the cultivation of the Artemisia annua plant, which unfortunately has a 10 to 12-month growing cycle. This long growth cycle resulted in huge fluctuations for this drug in terms of supply volume and hence pricing. The Gates Foundation hence decided to fund the development of a commercial scale process where Amaris developed a yeast-based fermentation process that produced artemisinic acid in 2 weeks, which could then be converted in a few weeks to artemisinin using chemical synthesis. The result of this project was stabilization of the price and supply of artemisinin on the market, which was extremely important for developing countries, especially in Asia and Africa. This was, of course, a non-profit project for us. Sorry, that was a little slow. The result of this project was stabilization of the price and supply of artemisinin. Now, this was, of course, a non-profit project for us, but we were able to apply this technology to other products as well. We are now focused on 3 main markets, the first one of which is clean beauty, and that includes our Bioscience brand, as well as products that use a squalene ingredient. I'll talk a little more about squalene in a couple of slides. But we sell our Bioscience brand directly to consumers through Sephora, as well as our own website, bioscience.com. We also sell squalene to other businesses through our distributors, as highlighted on this slide. The second market for us is in health and wellness. This includes multiple vitamins that are derived from fermentation and our soon-to-be-launched sweetener, as well as human nutrition ingredients, which includes our applications in the pharma space as well. Our partners in this market include DSM, Givaudan, and Nenter. Finally, the third market that we operate in is in the flavor and fragrance market, where we work with companies like Firmenich, Givaudan, and Takasago. In each case, these companies have a well-established presence in the marketplace for these ingredients, but are trying to solve some supply chain issue, whether it be cost, sustainability, or purity. And in every case, Amaris is able to perform all the R&D required to develop the strains and a new manufacturing process, manufacture the ingredient, and finally deliver the same product that gives our partners an advantage in their marketplace. So what advantages does an Amaris produced ingredient offer to its partners? First, because we can tailor the strain and fermentation conditions precisely, we can produce the same molecule at much higher purity than what might be obtained from a petroleum or plant or animal source. Secondly, we can manufacture the same molecule at a lower cost so that the benefits of a sustainable product aren't just available to people who can afford them. We believe that high quality products should be accessible to all consumers. And finally, fermentation is simply a better process than current manufacturing practices. which include the use of petroleum feedstocks or involve the harvesting of ingredients from plants or animal sources. Let me walk you through some examples that will help illustrate these points. So let's start with an example of a better product and better purity. Many of us have used stevia as a sweetener. While it works really well as a sweetener, there's no escaping the bitter aftertaste that is synonymous with stevia. The reason for the bitterness is that the extraction process for the sweetener results in a mixture of molecules, out of which the active ingredient, Reb M, is only 1% of the entire mixture. The rest of the molecules in the mixture result in the bitter aftertaste. At Amaris, we have developed a fermentation process where we are able to precisely tailor the strain and the process to make greater than 95% pure Reb M, resulting in a very clean, flavor-free, free from any aftertaste. Let's look at an example of better cost. The production of vitamin E has for decades relied on long and complex chemical synthetic route. Amyris produces a molecule by fermentation called farnesene, which can be converted to multiple products. Our partner Nenter figured out a process where the fermentation-derived farnesene can be converted to vitamin E using a far simpler chemical process that reduces manufacturing costs by 35%. Finally, a few examples where solutions developed by Amaris are simply a better process for the planet. We produce a fragrance ingredient for Firmenich, where the original source was a native plant, and 1 acre of the native plant produced about 6 kilograms of the fragrance. The same acreage of sugarcane results in 400 kilos of the identical molecule. We see similar results for a molecule that we produce for Givaudan, where we see at least 200-fold efficiency in land usage. Finally, we can't talk about sustainability unless we talk about squalene. So squalane is a molecule that is quite ubiquitous in cosmetics as an emollient. Squalane is produced from squalene, which is a biological molecule made by most living cells. Till about 8 years back, most of the squalene for the cosmetics industry was extracted from shark liver and converted to squalane. Every year, 3 to 4 million sharks were killed for this market. Amaris developed a process where we were able to convert fermentation-derived farnesene into squalene and were able to achieve purity that was the same, if not better, than shark-derived squalene. By our estimate, 1 square meter of sugarcane saves about 10 sharks, which is a testament for how biotech can offer a truly better solution for our planet. To date, Amyris has manufactured 7 different molecules at scale and currently has 12 other molecules in commercial development. Our goal and track record has been to commercialize 2 to 3 molecules every year. So how did we achieve this kind of track record? Well, with a lot of investment in R&D. We invested close to $500 million in developing a world-class strain optimization and process development pipeline by focusing on 3 main areas. The first is cellular programming, where we have optimized the process of assembling large DNA cassettes and integrating them into microbial genomes in order to introduce new functionality into the production organisms. We have also developed novel techniques to introduce entire pathways into a strain in a single transformation step. We also apply classical methods like random mutagenesis and adaptive evolution to find new strain lineages. Overall, we send about, 1.2 million strains per year for analysis, which then highlights the next area that we had to spend a considerable amount of time and effort in developing, and that is analytics. It's of no use to create so many strains if you can't measure the impact of the cellular changes. We have hence developed methods to not only analyze the extracellular molecules produced by the cells, but to also quantify the intracellular metabolites and proteins. We believe that it's important to not treat the cell like a black box, but to instead use it to expand our understanding of cause and effect so that the next molecule we work on can benefit from the lessons learned of the previous one. Our current pipeline can analyze to the tune of about 10,000 samples a day across multiple measurement platforms. Finally, as you can imagine, building so many strains and analyzing so many samples results in a huge amount of data that quickly becomes overwhelming for any human. We have hence developed sophisticated computational metabolic models that we can overlay with the data. We apply proprietary statistical models and machine learning algorithms to these metabolic models to understand the impact of the cellular changes and to drive the next set of decisions. As a data set increases, it will drive the next set of improvements faster, resulting in fewer and fewer iterations through the strain engineering cycle. All this investment has culminated in a fully integrated strain optimization pipeline where we combine next generation tools in molecular biology, metabolic engineering, and fermentation process development with automation and computational tools. However, what's missing with this picture is a final stage for this pipeline, and that is manufacturing. Because of Amaris' experience in manufacturing 7 different molecules with varying physical and chemical properties, Amaris has learned some hard lessons in the difficulties of scaling lab-based results to predictive performance at the manufacturing scale. Unless your entire R&D machine takes into account the final manufacturing requirements of your strain, even the most sophisticated pipeline will fail to deliver the final strain and process that performs as required at scale. Finally, we rigorously capture data gathered at every step in this pipeline and feed it back into the design process with the goal of continuously improving the pipeline and our understanding of strain physiology. I also want to call out the fact that we shouldn't discount the impact of human intelligence in this step. So while we do apply a huge number of computational tools to understand what's going on with the strain, we also have humans who curate this data and then try to understand what they should be doing next with this. I would now like to spend the next few minutes discussing some of the computational tools that we now apply to elucidate the most optimal pathway to a certain molecule, and design tools that enable scientists at Amaris to rapidly and without errors come up with thousands of DNA designs to convert an ethanol-producing yeast strain to, say, a farnesene-producing one. So as this slide illustrates, all living cells consume sugar in some form or the other and convert the sugar into all the molecules they require for survival. In order to hijack the cellular machinery to produce a target molecule, you hence need to first figure out the reactions needed to convert sugar to, say, a molecule like farnesene. We had then built a database or living atlas of all the reactions known to occur in living cells. We have developed tools that can access these reactions and for a given molecule map out the most optimal route to that molecule from sugar. However, this is not as trivial as I just made it sound. Imagine you use Google Maps to get directions from one location to another. The software will try to get you the most optimal route by avoiding major traffic bottlenecks or without sending you in the wrong direction down a one-way. In a similar fashion, our design tools can't just map the route with the smallest number of reactions. They have to take into account elements of thermodynamics, cofactor usage, and pathway balance to come up with the most optimal route from sugar to that final target. And so that's what this will look like. So in this case, for every, so what's important here is that the software needs to calculate what the highest possible yield from sugar to that molecule is going to be. So in this case, for farnesene, for example, for every 100 grams of sugar that is fed, about 24 grams is converted to the product. This is an important number because of what our manufacturing team does with this information is they combine it with all the operating expenses data collected from our manufacturing sites and develop a cost model to predict what kind of manufacturing costs we can achieve at commercial scale. So again, unless a company has experienced a full-scale manufacturing campaign, it's very difficult to understand just how much work the R&D team has ahead of it. Okay, once we identify the route, it's time to convert it into DNA code. Every reaction is carried out by a protein. Everyone knows this in the field. So every reaction is carried out by a protein. Every protein is encoded by a unique DNA sequence. Our design software will map out these DNA sequences associated with each reaction and combine these DNA sequences with the other DNA supporting elements like promoters, terminators, tags, etc., which are required to functionally express the required proteins. Of course, it's very rare that a single DNA code will provide the functionality you need, and typically you will need multiple DNA commands to program the cell to carry out the new activity. This is, of course, manually very labor intensive, as anyone has ever done this will attest to. So what we've done is developed a genome specification language for our scientists so that a simple written instruction like the one shown here translates into a complex DNA sequence as shown here. Now, for anyone who's ever done this, you understand what goes through this process. It's a process that a few years back required hours of manual curation, a huge amount of copy and pasting from public databases into Word or Excel. It's a highly error-prone process. Today, it takes a few seconds. Now, even though our understanding of biology keeps getting better and better, I would argue that biology remains highly unpredictable. And even picking the right DNA sequence for a particular reaction is not a trivial exercise. So scanning as much diversity as possible for every reaction is key to finding the optimal solution. And why is that? Again, these are various reasons that I have listed here. I'm sure most of you could come up with multiple other reasons why you need to scan genetic variants. But the top two, I'll just talk about the top two. You can read through the others. The first one is what happens if the native reaction doesn't exist. So for a lot of times, the molecules we're trying to make are not made by the production host that we're trying to use. It's also possible that the native reaction does exist in that host, but the activity is just too low. And it is possible that the same reaction is carried out far more efficiently by a protein found in another organism. Another reason could be that in order to get the activity you need to manipulate protein expression to increase the activity. You can do this by varying the codon usage. Now, of course, all the other reasons, decoupling the pathway from regulatory mechanisms, minimal side reactions, cofactor dependency, protein localization, all these are factors that you have to take into account. However, the only way to bring together this level of diversity in a host is by synthesizing the various genes. And this is where gene synthesis technology pioneered by companies like Twist have changed the landscape. How quickly you can make progress in any project is defined by how many shots on goal you can take in the experiments. And so over here, I show the design, build, test, analyze cycle that everyone goes through when we have to optimize a strain to make a single molecule. Now, like I said, how quickly you can make progress in this design, build, test, analyze cycle is how quickly you can go through it and how many times you can do it per unit time. The more high-quality shots you can take, the higher your chance of actually scoring. The cost of taking the shot, the amount of time it takes, and the quality of that shot all impact the likelihood of success. Now, I'm sure a lot of you can attest to this, but back in 2003, I used my first synthetic gene. It cost me $3,200, or actually cost my PI $3,200, and it took 3 months to synthesize a gene that contained multiple errors, which I then had to correct later on. I had to pick one candidate for this reaction, and I had to be either absolutely sure or really lucky that this was the right gene to pick. Today, if I had to do the same, and I had $3,200 to spend, I would be able to test 20 candidates in 1 month without worrying about the quality of the experiment. And this is how Twist has changed the game. You don't need to try and pick the right candidate. You simply test them all and reduce the chance that you missed anything. Now, let me show you some actual data from a screening experiment we ran at Amaris a few weeks back. We were working with a 7-step pathway and screened multiple genes for step number 5. As you can see, the results vary significantly from one version of that gene for the same reaction to the next. If I had only one gene to test, I would need to be incredibly lucky to not pick gene 4 or to pick gene 10, which are the lowest performers and the highest performers in this data set. But this is the advantage that cost-effective, high-throughput gene synthesis brings to companies like Amaris today. It takes the guessing out of the equation. You can test a huge amount of variation and pick the right one based on actual empirical evidence. Now, this is where a company like Twist really gives us an advantage. Now, let me finish up last few slides by tying all this back to our ability to predict performance at manufacturing scale. And this is by focusing on data quality and prediction of strain performance at the lab scale. When you need to test 1.2 million strains a year, you can't possibly test them all in fermentation tanks. And yet, that is exactly what you need to do to predict manufacturing, a manufacturing process. What we've done instead is we've developed culture models in 96 cell plates and used them as surrogates for predicting performance at the larger scale. understanding fully well that there are differences between the 90 square plate model and the fermentation and the half-liter or 2-liter fermentation scale and the manufacturing scale. In order to account for these differences, what we've done is develop algorithms that pick winners from the plate models that are then tested in lab-scale fermenters under conditions meant to reproduce what the strains will experience at manufacturing scale. Again, because of our experience in scaling up strains and processes, we've developed a lab-scale fermentation pipeline that is capable of perfectly predicting performance at manufacturing scale. This graph represents different strains for one product that was sent to manufacturing over the years. And in every case, the lab performance has matched performance at manufacturing scale. This level of reproducibility dramatically reduces risk at manufacturing, where every error is risky. And this reproducibility also shortens time to market. Let me now show you a quick video. Let me show you a quick video of what a large-scale fermentation facility looks like. This is a fermentation facility that we operated in Brotas, Brazil. It sits next to an ethanol mill that we buy sugarcane juice from. These pipes you see are transporting the crushed sugarcane juice from the ethanol mill over to us. And let me see if I can go back a little. And here you can see the crushed sugarcane, which is known as bagasse, piled up here. This bagasse is burned to generate electricity that runs the entire facility and also supplies power back to the grid. This is a bird's eye view of the 6 fermenters along with the storage tanks, chillers, and compressors. So we have 6 fermentation tanks at this facility. Each fermentation tank has a volume of 200,000 liters, resulting in a working volume of about 1.2 million liters per fermentation run. We also utilize multiple contract manufacturing groups to accommodate the different molecules that we produce, and are currently in the process of building 2 new manufacturing facilities as well. So, in summary, over the past 10 years, what Amaris has done is built the first fully functional automated organism engineering pipeline. This has resulted in a 10-fold lower development cost for new molecules and a 5-fold reduction in development time. We have successfully, to date, commercialized 7 target molecules and produced them at the kilo to ton scale. We currently have 12 molecules in the development pipeline and expect to launch 2 to 3 molecules every year for the next 3 years. And I think finally, it's important to note, low-cost, high-throughput, high-quality DNA synthesis has been a game changer for the industry, reducing risk and uncertainty in optimizing strain performance. So with that, I would like to thank you for your attention. I'm going to now hand it over to Christy.
Thank you, Dr. Chandran. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'll conduct the Q&A portion of the webinar after the presentations have concluded. And next up is Mr. Downing. Please go ahead.
Thank you, Christy. My name is Jason Downing. Thanks, everybody, for taking the time to join the webinar today. I have a brief presentation that'll give you a bit more insight into how Twist's technology is powering the synthetic biology and genomics revolution, as you just heard from Sunil. I also want to take a moment and thank Sunil and Amaris for giving such great insight into how they are using Twist technology to fuel growth and expansion in new applications, new discoveries, and just take a moment to provide some insight into how our technology is really driving these discoveries and making scalable science happen. So as many of you probably know, the principles of classical gene synthesis is where you take approximately, say, 100 oligos. You're going to assemble those oligos and then clone this into a vector. This classical method is the foundation of many DNA synthesis platforms that exist today. For instance, using about 56 40-mers, you can produce about a 900 base insert or gene. However, looking at the 2 figures that we have in front of us, you can see that scaling this, especially to larger genes, inherently with existing technologies before Twist, adds time, it adds cost, and it adds quite a bit of complexity, creating significant limits to feasibility. This is really where Twist comes in, and where we're really changing the game for companies like Amaris. Twist's silicone-based synthesis platform provides researchers with the ability to move beyond what was previously possible. Traditional-based methods rely on standard laboratory plates, like what you see up in the top left. One 96-well plate of oligos can produce approximately one gene. While effective, this method is generally slow and expensive. It's very difficult to provide cost-effective solutions using this type of synthesis. Really goes to what Sunil was talking about, how you'd have to get lucky to really find the gene that you're looking for, and you can't really do that design, build, test cycle anywhere near as effectively as what they've been able to do. There's also array-based oligosynthesis. These methods provide good flexibility, but the quality of data generated is pretty low. In addition, array-based methods suffer from high lot-to-lot variability, which can pose a lot of challenges depending on the application that you're working with. With Twist, Twist synthesizes the probes using traditional chemical synthesis processes, which have been around for decades. What separates Twist from everyone else is the ability to scale that chemistry to very small reactions using our silicone-based platform that you see on the right-hand side. By combining cutting-edge silicone-based manufacturing processes with robust scalable oligosynthesis chemistries, Twist can provide researchers with both customization and high-quality data like never before. So when you look at the two in comparison, a 96-well plate produces one gene and it takes quite a bit of time and cost, definitely not scalable. With Twist platform, one silicone chip can produce many, many oligos and generates 9,600 genes, a drastic step function difference from where we were previously. So now that we've kind of thought through the foundations of Twist technology, I wanted to dive a bit more into Twist gene synthesis portfolio, just kind of give you an idea of what's out there. So with this portfolio, we offer a suite of products for your synthetic biology needs. Our scalable DNA synthesis technology allows you to create genes up to 3.2 kb, and as Twist, we are always pushing our technology further. The scalableness of this allows you to produce one gene or produce 100,000 genes up to 1 million genes. So it's really integrated well into the scalability of the design, build, test cycle that Sunil was talking about. Regardless of the application, be it drug development, gene editing, pathway assembly, just to name a few, the whole point for Twist is to scale to your needs without introducing quality issues or causing it to be too costly. In terms of what we have to offer, the Twist platform is very flexible and scalable. So now you can really envision limitless genes and unlimited discovery. We have easy ordering through our e-commerce platform. I would definitely recommend, if anyone's interested, take a look at that. We have no minimums for our genes, and we have very competitive pricing in the market to make sure you can scale and not break your budget. Our portfolio has everything from gene fragments and non-clonal genes to having your gene of interest cloned perfectly into one of our off-the-shelf vectors or your vector of interest. So with that, I will let us get to questions, but the only question I have left for you is, what can Twist build for you? Thank you, everyone, and Christy, I'll turn it back over to you.
Thank you, Mr. Downing. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. All right, we'll now start the Q&A. Sunil, the first question is for you, but Jason, feel free to chime in if you'd like to. It seems that several companies and institutions are trying to develop fully automated biofoundries. So how far do you think the industry is from such a biofoundry, and what do you think the biggest challenges are to getting there?
Yeah, I mean, great question. So yeah, there are companies, there are other institutes that are trying to do the same thing as Amaris have. I'll speak to what we have been able to do. So we have a fully operational biofoundry. So in terms of the question, yes, Amaris already has a working biofoundry that we've been using for the last few years. The scientists at Amaris, I think they appreciate it because, instead of moving liquids around in the lab, they're actually reading papers, analyzing data, and trying to figure out what the next hypothesis seems to be. In terms of the second part of the question, the challenges, I think the biggest challenge, one of them is going to be, what host do you onboard into a biofoundry? So back, in 10 years back, everyone worked with E. coli or Cerevisiae as a host of choice. to produce any new molecule. But today, that thinking has changed. Today, the question is, what's the right host for that molecule? But of course, onboarding a new host to the point where you can do 1,000 transformations a week, which is what we do at Amaris, that's not easy. So that onboarding process is always going to be a challenge. The second one is, again, what I alluded to in terms of manufacturing. You can have a world-class biofoundry. You do need to understand where that strain, the final strain that you develop, is going to be going. What's your manufacturing process looking like? What kind of stability constructs do you need? So those are all inputs that you have to figure out on day one. So I didn't spend a lot of time on this, but in our automated design, right up front, we include a lot of DNA constructs, knowing what the manufacturing process might look like, and so we account for some of that variability right up front in the automated design side. So yeah, I mean, the companies and institutes coming along. Amyris has a fully functioning one already. It keeps improving. Of course, there's a lot more work to do.
And Sunil, just to clarify, is your biofoundry fully automated?
Yeah, so certain parts of it are. So fermentation, of course, is not easy to automate. We have automation tools for sampling from fermenters. The biology side, yeah, most of that is fully automated. I would say more than 95% of our DNA assembly is now done by the automated pipeline. Now, we do have a team of scientists that operates that pipeline. So, I'll be careful. It's not like after we just have robots doing all the work on their own. We have humans supervising all these operations.
Great, thank you. And Jason, anything to add to that?
Oh, nothing for me. That was excellent.
Great. Sunil, with multiple genes using the same promoter or terminator combo, have you ever been concerned about genes recombining with each other? And is there a limit to the number of genes using the same promoter-terminator combo for genome stability of the final strain?
Yeah. Sounds like a question from someone who's actually lived through that. Yeah. No, you have to be really careful with reusing the same promoter and terminator. So what our design tools look for are repeat elements within the same DNA assembly. So there are a couple of things to look for. When you're assembling your DNA, you need to make sure that within that same assembly, you don't have similar DNA parts that could essentially cause a loop out event. You also have to make sure that the different assemblies going into, for example, the same chromosome of yeast don't have similar elements that might cause that pathway to loop out. So yeah, it's definitely a concern. We are careful with how we reuse our DNA parts. The other part I didn't really talk about much is we sequence everything that we make over here. So all the DNA assemblies we produce are sequenced. So we sequence about 6,000 plasmids every 2 weeks using our sequencing pipeline, and we sequence about 8 to 10 genomes a day to make sure we catch any stability or instability events. Good question, though.
So after mutagenesis, have you ever had an occasion where you found an interesting mutant and then characterized that mutation in detail? And specifically, do you determine if this promising mutant leads to changes in protein expression versus improved function of the protein itself?
Yeah, exactly. Yeah, so the mutagenesis pipeline, we introduce about, in every mutagenesis cycle, we introduce about anywhere from 30 to 40 mutations per round. We'll then screen them, pick for performance. The winners are then sequenced, and we figure out exactly every mutation that's happened in that strain. And then what we'll do is we will take each mutation or a couple of the mutations in combination and go back to the original lineage and put them back in and try to find the causal mutant in there. And then once we find the causal mutant, we then try to study, okay, what was the reason that mutation caused a benefit in our yield? So is it, like you said, is it a protein function issue, or is it solving for protein expression, or is it solving for regulation? But these are all experiments we do. So yeah, like I said, because of the throughput we have in this pipeline, we are able to sequence most of the winners, and then go back and recapitulate the mutations in a clean background.
Sunil, as the cost has dropped and the availability of synthetic DNA has increased, how has your approach to microbial engineering changed?
Yeah, I mean, I think I mentioned this during the presentation. We are able to try out a lot more ideas than we were able to in the past. So, instead of just testing 1 or 2 gene variants, we'll try a bunch. I mean, we just had a project recently, where we essentially screened, I think, about 75 gene variants for a single step. The other issue that I didn't talk about much, I alluded to a little, is codon optimization. And originally we always thought that if you optimize the codons once, that's more than enough. But then we realized slowly that different codon optimizations for that same gene make a huge difference in the protein expression. So what we've done is try to reduce that uncertainty, and that's what low-cost DNA synthesis has allowed us to do. Now, of course, you do need a biofoundry that's able to handle that kind of throughput, because having 300 gene variants is great, but how do you assemble all of that?
And how often are synthetic promoters and terminators used for strain optimization, and how have synthetic promoters and terminators impacted overall yield?
Yeah, that's a great question. So synthetic promoters, we found synthetic promoters to be very useful because we can decouple native pathway regulation from actual pathway expression. So one area where we do use, I would say it's a semi-synthetic promoter, because really we cobbled together a design based on natural designs that we found out there, but it's still a novel promoter. We use it to actually stabilize the strain. So one thing I didn't talk about is that manufacturing, very early on, we found the strains to start becoming unstable. So the yield and productivity would start dropping, let's say after 8 days in a fermenter. And we started realizing that it's the cell's strange way of essentially fighting back, because you're trying to force it to produce a molecule that it doesn't really want to produce. And so these stability promoters help us carry the fermentation a lot longer and then maintain a very high yield and productivity, which of course then reduces the final cost of your production cost of your molecule.
Sunil, can you tell us if you use next-generation sequencing to characterize mutant libraries, and if you do, what that pipeline looks like a little bit? And then to follow up on that, what software do you use to quickly analyze sequencing data to find interesting mutants?
Yeah, I'll talk about the second part first. So unfortunately, when we started developing a high-throughput sequencing pipeline, we were unable to find any software out there that could handle that kind of throughput. So again, we were sequencing 6,000 plasmids in a single MySync run. We were essentially doing a lot of multiplexing and barcoding, and then deconvoluting the data at the end of the sequencing run. So we ended up writing our own software. Some of the details on this I think we did publish a paper describing our sequencing platform for plasmids, I think 2 or 3 years back in ACS Synthetic Biology, but it is proprietary. Now, the first part of that question about sequencing our mutant libraries, yeah, we do. Again, it depends on the project. Some projects need us to sequence everything they produce. Some of them ask us to sequence only a few. It varies. I can't go into details on how we do it exactly, but again, you can sequence anywhere up to 8 full genomes in a single run in a day. And if it's a library, we can of course do a lot more. But we do. We have access to multiple sequencing platforms here at Amaris.
Sunil, we have a question going back to when you spoke about codon optimization. There have been a number of papers that have challenged the value of CAI, the most used codon approach. Do you have a proprietary algorithm to perform codon optimization, or do you vary based on some version of CAI?
No, we have our own proprietary version. It's based on data that we generated in-house. And so, I mean, really what we did is we sent out a huge number of codon-optimized proteins based on different algorithms that we had written in-house. The different genes came back. We analyzed the expression levels, looked at the data, and then refined the algorithms and back and forth. So, no, today the algorithm is proprietary to what we do. It's based on a lot of our data that we generated here.
Okay, great. When integrating engineered genes for desired traits, which approach do you think has the greater success? Is it directed or random mutagenesis?
That's a good question. It's mixed. I would say anywhere from 20 to 30% of our gains in certain projects have come from mutagenesis. But sometimes, mutagenesis is not going to give you, for example, a protein activity that did not exist in that host. I mean, it might, but the chances are really low. So no, I mean, for us, we have to do both. You can't rely on a single approach and hope to get to the yield and titers you need. So, but yeah, about 20 to 30% of our yield gains have been from mutagenesis, but the rest have been from integrating new pathways into the genome to get the trait you need.
And how many different organisms do you typically look at when approaching a biological target?
Well, we'll always start with, okay, are some of the common industrial hosts, are they viable candidates? So we go through the normal, is it going to be Cerevisiae, is it going to be E. coli, is it Pichia? And so we will do that analysis on day one before we even start the project. But I would say we look at anywhere from 5 to about 15 different organisms. But again, the challenge is that you have to weigh the cost benefit of picking a brand new host for which very few tools exist, because you have to factor that into your R&D timeline, versus picking a host like, let's say, Cerevisiae or Pichia, which may not be perfect for the molecule, but with the tools that we have today, you can make progress a lot faster. But yeah, it's not a huge number. It's not 100. It's still a number that's manageable.
Regarding directed evolution, do you use any homology models or modeling of the protein structure to target specific residues?
We do. I mean, as long as the crystal structure or at least something is known about that protein or related protein, then yes, we do. We do apply. We do look at residues that we might want to engineer. A lot of times, proteins that we are trying to improve, unfortunately, don't have any structural data around them. In that case, we have an enzyme engineering pipeline, which I didn't talk about much at all, where we end up screening a huge number of enzyme libraries to find the right one, and then start combining the different mutations based on a rational approach.
And do you have a specific strategy that you use when a coded protein is not soluble?
Yeah, that's a good question. When it's not soluble, so one is, scan for diversity. Again, find a protein that is soluble. So sometimes a protein might be membrane bound, or it might be localized in mitochondria, for example, and you need it in the cytosol. So in that case, try to find a version that does work in cytosol and is soluble. The other one is codon optimization. Again, we found cases where different codon optimization for that same protein changes its solubility. Third one is tags. We use certain tags that, I think most of you have probably seen these in literature, but we use different tags to try and solubilize the protein when it's expressed inside the cell.
Jason, we have a question for you. How does Twist technology control complex gene sequences such as poly-N runs or triads for oligo and gene synthesis?
Great question. So our technology up in front of it, as I mentioned, our e-commerce platform, has embedded in the design algorithm for gene sequences that would move into our production pipeline. That's kind of a first step, and if things prove challenging for that, we have excellent technical support and bioinformatics support to partner directly with customers on those challenging sequences to figure out what the best strategy is to be able to get your gene of interest.
Great, thanks. Sunil, when engineering a pathway, what's the typical number of enzymes or variants that you try for in every step?
Oh, it's all over the place. It depends. Like I said, recently we tried 75 different variants for a single reaction in a pathway. In other cases, you might not find a lot out there. Sometimes it's just 2 or 3 because that's all that you can find. And then you have to do, you have to essentially take a guess and start doing more diverse sequences that can be related to the reaction you want, but are not exact matches. I think the number is all over the place, really. It could be as, in one case, we tested greater than 100 variants for a single reaction. But it changes from product to product.
And how do you select targets of molecules for microbial production?
That's a fantastic question. So the way our model has always worked is we have our partners who have had the molecule that they've been selling in the marketplace. So they come to us with that target. What we do then is we look at a few factors. One is what is the cost target that the partner requires for that molecule? And then we look at the biological feasibility of making that molecule. Based on the biological feasibility, we run our models to see, okay, what kind of yields and productivities would you need to hit at manufacturing scale to meet that cost target? And then are those yields and productivities doable based on our experience over the last few years? So this is, again, where I talked about our experience in manufacturing. We have cost models where we can look at numbers, manufacturing numbers, and then make that call. So yeah, it's not trivial to figure out if a target molecule is doable or not. Sometimes it's biologically feasible, but economically doesn't make any sense to go after it. Sometimes economically it makes a lot of sense to go after it, but biologically it's just not a good target. So yeah, there are a lot of nuances, and with experience, you hopefully start picking the right targets.
And Sunil, are there any classes of molecules that you're just not willing to attempt to reproduce or to produce?
Sorry, could you repeat that?
Are there any classes of molecules that you're not willing to attempt to produce?
It depends on the market economics. I think in the end, there are 3 categories that Amaris feels a molecule has to fit into, right? What's the reason for making that molecule? Is it a sustainability question? So can we solve a sustainability problem with that? Is it a cost issue, or is it just purity? But it has to meet one of those criteria. And again, you have to be careful with some of the molecules that people want you to make. You do have to make that. You do have to be a good judge of what that molecule could be used for. So yeah, so we are careful with the molecules we select and that we go after.
How do you know when you're done? Is it when you've met a certain threshold or a target set by a partner?
Exactly that. So again, mentioned it a couple of times. On day 1, when we start a project, we know exactly what kind of performance metrics we need to achieve for that strain and for the process. And so every 3 months, we are monitoring, okay, where are we at in terms of things like performance metrics. And because we can accurately predict what the manufacturing cost is going to be based on experiments we run at the lab scale and the pilot scale, we are able to call a project when we hit that cost target set by the partner.
And what's the most impactful thing you've ever learned about target selection?
How much time do we have? No, that's a good question. I mean, there's certain molecules that just make sense to go after in fermentation. But sometimes you have to step back and look at the molecule and say, maybe a an entire fermentation process is not the way to go for this. Maybe you need to stop at an intermediate and then finish off that production using a couple of chemical steps. So it's not necessary that an entire fermentation process is the best process for the entire biological target. So, you know, not just because it's biologically produced doesn't mean you can make a successful fermentation process that meets cost targets. It's a lot more nuanced than that.
Has there ever been anything that you thought was important in target selection that turned out not to really impact your success rates?
So if I read back the question to you, so has there been an aspect that we thought was important but it's not important anymore? And let's think about that one a little. That's a good question. Not as much as I know. I think it's been the opposite for us, really, which is, over the years, there have been things that we didn't think of that came back to haunt us. And so, it's been more of that rather than, has there been something that we, thought about but didn't matter later on.
And can you tell us a bit about what's been your most successful program and what has been your least successful? Of course, considering those that are already public knowledge.
I mean, I would say every molecule that we've sent to commercial scale has been a huge success for us. Everyone has had its challenges, and we managed to solve for all of them. The reason I wouldn't call anything as the biggest success is simply because each molecule has had its own set of challenges. So some of the molecules we produced were solids, some were liquids, some were mixtures of liquids, some were amorphous powders. Every single molecule had its own set of challenges that we had to solve. So the learning opportunities, themselves made every molecule worth it to go after. In terms of molecules that, very early on in our history, there were certain molecules that we worked on that I can't really talk about, that we ended up, realizing later on that, okay, these would probably not make commercial sense to go after. This is when we were a much smaller company that didn't have a lot of manufacturing expertise. I think today, we've had a fantastic track record of taking molecules to market.
And how many modifications to the yeast genome are you able to make in a single engineering cycle?
Yeah, that's a good question. The most we've done is we integrated, I want to say it was 30 kb of DNA in a single transformation step. We've probably gone even as high as 50 kb. Now, is that something we do on a regular basis? No. But the pipeline is capable of handling that level of engineering and that was in Saccharomyces, where you can rely on a very good recombination machinery.
How do you identify and manage host toxicity and compound transport?
Oh, so compound transport is a good one. Again, because of the number of molecules we've worked on at Amaris, we have a pretty good idea on whether that molecule is going to get transported out of the cell or not. Now, a molecule not getting transported out of the cell is not a handicap in any way. It's just a question of what your cost target is. And there are plenty of molecules that don't get excreted, but which can be made quite successfully in fermentation. The host toxicity is a big one. And again, you can make molecules that are toxic to the host at a certain level. The question is, at what level does that toxicity manifest itself? So if it's at a level that's above where you need to be to hit the cost target, then you're okay. If it starts becoming toxic at a much lower level than you need to be to hit target economics, then you need to solve for that, and that can be a challenge. I mean, that's where again, mutagenesis or adaptive evolution really play a huge role, and we've done that in the past.
Great, thank you. And those are all the questions that we have in our queue. We'd like to thank Sunil, Chandran, and Jason Downing, and our sponsor, Twist Bioscience. As a reminder, please look out for the pop-up after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.